Yes.
Shall we start?
Yes, sir.
Okay. Good evening. Everyone, welcome to IMSC, Empowering People, Enhancing Education. This will be more of a online interactive session. If you have any questions, any doubts, you can unmute yourself and you can ask your questions. I'll be taking biochemistry. Today, our topic will be transcription. Let me introduce myself. I am Dr. Prajwal. I have 2 years of teaching experience in biochemistry. Okay, why should we know transcription? So, you all know basics like what is transcription, synthesis of, hello? Yeah. Synthesis of RNA from DNA is called transcription, and synthesis of protein from RNA is called translation. And all together, this is called as central dogma of molecular biology. And the whole molecular biology is based on this. A whole molecular biology is based on this. So what is RNA? What is actually transcription? Synthesis of RNA from DNA by RNA polymerase enzyme is called transcription. So what all RNAs can be synthesized? All types of RNAs are synthesized from DNA. So what all the types of RNA we have? Like we have protein coding RNAs and we have non-protein coding RNAs. Sorry for this one. Yeah. So we have 2 types of RNA. That is your protein coding RNAs and your non-protein coding RNAs. Your protein coding RNAs mainly includes your mRNA. That is the one which codes for your protein. That is what is called as translation. Whereas your non-protein coding RNAs includes your tRNA. It is transfer RNA. Your rRNA, which is ribosomal RNA. siRNA, which is silencing RNA, long non-coding RNA, and small nuclear RNA. So what are the functions? tRNA is your amino acid carrier in translation, and rRNA is your ribosomal RNA that helps in translation. And these 3, siRNA, long non-coding RNA, and small nuclear RNA helps in regulation of your protein synthesis. So here you have one important point to be remembered. rRNA is the most major component of all RNAs. It almost is 80%. This can be asked as a MCQ in your PG entrance examination. So which is the main component in RNA means that will be always your ribosomal RNA comprising of 80%. So are you able to get till now?
Yes, sir.
Okay. So coming to, there is a similar process like transcription, that is replication. You all know that synthesis of duplicate DNA or just DNA from DNA is called replication, whereas synthesis of your RNA is called as transcription, especially your mRNA. So both is like, both will use DNA as the synthesis and both will use different types of polymerase enzymes for synthesis of DNA in case of replication, whereas mRNA in case of transcription. But there are certain similarities and certain differences that you should remember. So you all know whatever that is synthesized will be in 5' to 3' direction. 5' to 3' direction. And both will have initiation complexes. We'll come to this in later part of the topic. What is this initiation complexes? And all will follow Watson-Crick base pairing. That is, your A will bind to T and your G will bind to C only with triple bond. So coming to differences, as the name suggests, RNA will have riboxynucleotides, whereas in replication, DNA will have deoxyribonucleotides. And you all know that in RNA, there will be no thymidine. Instead of thymidine, there will be uracil. So in case of transcription, there will be no T. So there will be uracil getting added opposite to A. And whereas in replication, if the DNA polymerase need to synthesize new strand, there should be some primer to start up. But whereas in transcription, you don't require that primer. So what is primer? It's like either it will be generally made of RNA that requires for DNA polymerase to start the DNA synthesis. And whereas in replication, your whole genome will be replicated. Whereas in transcription, the part of protein of interest only will be transcribed to mRNA. And this is question for you. Why you don't require proofreading in transcription, but you need proofreading in replication? So anyone can guess? Hello? Anyone wants to answer? Why there is no require of proofreading in case of transcription, but you require sophisticated proofreading in case of replication? Okay, what is proofreading? I have a question. Okay, what is proofreading? So we'll see. If this is a template strand, that is your DNA strand, and you are synthesizing a new strand. Let's take it is a new DNA strand is being synthesized. Once enzyme synthesize, it will recheck whether it has added exact nucleo. If this is A and this is G, it should add C here and it should add T here. So the enzyme will check whether it has added exactly T, whether it has exactly C or not. If it doesn't add, if it has not done correctly, it will go back and it will correct it and it will come back to its normal synthesis process. So now you have got know what is proofreading. So can you tell why there is no requirement of proofreading in transcription? Okay, I'll tell. So in case of replication, this happens before, generally it happens only before cell division. So that means it will duplicate whole DNA and it will be inherited to the next cell. So if there is any mistake in the whole genome, the whole cell will be like, the mistake will continues, that is your what do you call as mutation or misplacement of nucleotide will be continued for next generation. So all the proteins that synthesize from new cell will be bad proteins, or it will be wrong proteins that will be synthesized because wrong DNA has been synthesized during replication. Whereas in transcription, for one protein there will be lot and lot of mRNA will be synthesized. So even one mRNA goes mistake, other mRNAs will be taken, and it will happen only at one point of time. That will be neither inherited nor the mRNA is very long stable to produce more or more proteins. mRNA is not that stable, so it will produce less amount of protein. So if at all the mistake is happen also, it is limited to only less amount of protein. It is not for all the proteins. And also, whereas replication only happens once in the cell when it before it divides, whereas transcription will be a continuous process. If you want to proofread, it will be very time taking and energy taking. So that's why you don't have proofreading thing in transcription, but it is present in replication. Did you get this? Okay. Okay, we'll go to transcription proper. Before going to transcription proper, you should know few terminologies like template strand, promoter, initiation, termination, and everything. So first we'll come to template strand. So let's take, this is a DNA duplex. This is 3-5-N and this is 5-3-N. Okay, so let's take mRNA has been synthesized. I said already, whatever it is synthesized, it will be in the 5-3- direction. That will be in 5-3 direction. Okay, so the strand which upon against which mRNA synthesis is called as template strand, because it acts as a template based on which your RNA polymerase is synthesizing the new mRNA strand. So, and what is this strand? See the lower one diagram. If you can see, this is your template strand and this is your new mRNA strand that is getting synthesized in 5' 3' direction. So this is following Watson-Crick base pairing, and if you can see, there is similarity between, okay, they are not, one second, I'll draw a strand. Okay, if this is a duplex, what will be opposite here? A, G, G, T, C, G, and T. Okay, this is your opposite strand. If you see this opposite strand and your mRNA, it almost similar except for U and T, where T is replaced by U. So it resembles the coding strand which codes for protein. So that is why called the non-template strand is also called as coding strand, and that strand actually makes sense. That means it makes sense. That means it actually resembles the RNA transcript. That's why the non-template strand is called as sense strand or coding strand. And there is no compulsion that this strand should only act as template strand. This strand also can act as template strand in this direction. If you can see this image, the lowest strand is acting as template strand for gene A, gene B, and gene D, whereas upper strand is acting as a template strand for gene C. Did you get this? What is difference between template and non-template strand? Now, coming to next, we have seen what all required. One is you need a DNA template for synthesis of RNA. Next, we need an enzyme, and the other one thing is your nucleotides. So we'll see how the enzyme functions. So whatever the research has been done, everything was basically done in prokaryotes, mainly in E. coli, and in eukaryotes, it is mainly done in yeast cells. The structure presently I'm showing is of prokaryotes, that is from E. coli. So what does it contain? It contains mainly 5 subunits: 2 alpha, 1 beta, 1 beta dash, and 1 omega. So all the 5 subunits together is called as E. So this is the main component of RNA polymerase. Once this sigma factor binds, it gets activated. And this sigma factor has 2 functions. One, it increases the affinity of binding of RNA polymerase to particular site, and it decreases the affinity from binding to other sites. What is this particular site and other sites? I will come in next slide. So this is how your prokaryote works. In RNA polymerase is shortly called as RNAP, or you can call it, when it is activated, it is called E sigma. So coming to eukaryotes, eukaryotes also have similar structure, whereas in your prokaryote it is somewhat smaller inside, it about 450 kilodaltons, whereas in eukaryotes it is around 600 kilodaltons. So what is they also have 1 beta and 1 beta dash unit. Along with they have some small subunits around 12 to 14 it will be present. It depends on various types. Eukaryotes have 3 types of polymerases: type 1, type 2 RNA polymerase, and type 3 RNA polymerase. So how they differentiate this type 3 is based on sensitivity to protein called alpha amantine. This is a toxic protein that comes from mushrooms. This is also asked as MCQ. How you differentiate type 1, type 2, type 3 polymerases based on which protein toxin, that is your alpha amantine. So how it differentiates? When you add this has no effect on type 1, whereas it has highest effect on type 2. That means it totally inhibits type 2 polymerase, whereas on type 3 it has intermediate effect. And this type of RNA is mRNA. So which polymerase synthesize mRNA is also very important MCQ. That is type 2 RNA polymerase of eukaryotes synthesize mRNA. There are no types in prokaryotes. Okay, let's move. Shall I move on to next slide? You have any doubts till now? Okay.
Yes, sir. Clear, sir.
Okay.
Clear, sir.
Coming to how a RNA polymerase knows to synthesize which protein and where should it start and how it should start. So what happens? Before, let's think now, just genome is there and cell thinks these proteins are some signal it gets from outside of the cell. So something called transcription factors will be there in the cell. What are these transcription factors? The name itself says it's related something to transcription. What does it transcription factors does? So transcription factors have specific sites to bind. It will not bind wherever sequence it is there. It will have certain sequences. So each proteins will have different types of transcription factors. So these transcription factors will go and bind a particular site called promoter regions. Promoter regions. What does this promoter regions do? Promoter regions is where this transcription factor binds your RNA. Once transcription factor binds, your RNAP, that is your RNA polymerase, will be always scanning your whole genome. It will be always scanning, continuously scanning your whole genome. Whenever it sees a transcription factor binding to a promoter region, it will come and bind to that site and it will start synthesizing your mRNA. So how big this is RNA polymerase? It generally occupies around 40 to 70 nucleotide space it will occupy. That means if it binds here, it will come till to the 70 nucleotides. So we can't, so for our easy notation, that is for our easy understanding, exactly where mRNA synthesis, that is first nucleotide of mRNA, we named it as +1, and downstream we name it as +1, +2, +3 sites, and upstream we give it as -1, -2, -3, -4. Why we should name means that for our easy understanding, if we are doing some research study or we are doing some gene therapy things, we should know where exactly you are targeting. So for our easy understanding, you will use these notations. So where to start, where to end. And this +1 site is called as your transcription start site. The upstream is called as transcribed region. That is where actually your mRNA is synthesized, and the downstream is called as transcribed region, and the upstream, that is above, where actually the regulation is happening, is called promoter region. In prokaryotics, there is this sequence, either Tata sequence or TGTT GACA sequence. These are the sequences that will be recognized your nuclear factor and your RNAP, that is your RNA polymerase enzyme. So once this sequence is recognized, it will bind to that sequence and we'll see what happens next. And hold together this coding strand, promoter and RNA polymerase, all three together forms your transcription unit. So what happens in eukaryotes? Eukaryotes has somewhat complex regulation and somewhat complex transcription units, that is start site. What actually different? I will simplify. You should know first what is basal expression and what is regulated expression. can anyone tell what is difference between basal expression and regulated expression? Hello? You should also speak. This is an interactive class. If you know, you can tell. If it is wrong also, it is not a problem. You can speak. Okay.
You can ask them, sir.
Yeah. Hello.
Yeah, Anudeep, please answer.
Sir, Sir, I don't know.
Yeah, you can tell.
Sir, I don't know actually.
Okay, you don't know. Okay. Okay.
You can ask them by name, sir. In attendees list,
Okay.
their students' names are there. You can ask calling by their name.
Okay. Okay, got it. Okay, I'll tell what is basal expression and what is regulated expression. See, cell at normal level, maybe cell at when it is normally functioning, it will be requiring few proteins. That is, by default, they will be requiring few proteins. For these proteins, there will be no regulations. So it should be continuously synthesized. So that means there will be basal levels that will be continuously synthesized. That basal levels means normal function. At normal function, it will be continuous. That means actually there is no regulation. It will be continuously synthesized. That is basal expression. Whereas regulated expression, if cell goes to some work or goes to some stress or functioning its own functions, it needs to increase these proteins in amount. So that's where comes regulated expression. Sometimes it may need to decrease the protein synthesis, sometimes it may need to increase. That's where it is called regulated expression. So if that's where regulated process. And we saw in prokaryotes, the promoter side was in upstream, that is in minus regions. Whereas in eukaryotes, it is also present up till few nucleotides even in downstream. If you can see, this is a pleasant one. This is also a promoter region. This too is also a promoter region. So promoter region, so what it means, it occupies both downstream upstream regions and downstream regions. This is one major difference from prokaryotes and eukaryotes. And also there will be basal regulation, regulated regulation in eukaryotes. So how there is no much of basal expression that will be constantly expressed, whereas in regulated expression there will be something called as enhancers and repressors. The enhancers will increase the expression, whereas repressors will decrease the expression, and few other regulatory elements may also participate in the regulated expression. So did you get now what is difference between basal expression and regulated expression?
Yes, sir.
I heard someone saying I don't know. Did you get that?
Yeah,
Yes,
yes,
sir.
sir.
Yes, sir.
Okay. So
Yes, sir.
now we know promoter, we know RNA polymerase, we know what is template strand. So we'll now go into proper transcription. This is your transcription cycle. We'll actually transcription cycle has only 3 steps. This all together comes under initiation, and next is elongation, and next is chain termination. As the name suggests, initiation means once the mRNA synthesis is started. Elongation is that continuously adding of nucleotides till the process is completed, and termination is separation of newly synthesized mRNA from the DNA is called, when it happens, it is called chain termination. So we'll go into each step. So this is your DNA strand, double strand. So once your RNA polymerase recognizes your promoter site, it will bind to the DNA, and I have said that our DNA polymerase will occupy around 40 to 70 nucleotides. If you can see, it is occupying around 7 rounds of DNA. That is, each round, carries, each turn carries around 10 nucleotides. So it occupies around 70 nucleotides. You can see it in the here. So once it recognizes, it binds to promoter site. So once it binds, this is called as your template binding and closed complex formation. That is, still your DNA is in closed structure. So once it binds, it will use helicase enzyme that is present in the RNA polymerase to unwind the DNA template. So once this unwinding started, the point at where the unwinding started is called as transcription bubble. And once it is done, it is called as open complex formation. So in first step, what happens? As I told, from +1 site, your mRNA synthesis will start. Let's assume this is your template strand. So first the first nucleotide will be bind. So if this happens, it is not called initiation. Once the second nucleotide bind and there is formation of phosphodiester bond between these two nucleotides, then it is called as initiation. Then it is, now you can say that transcription has been initiated. So what is transcription? Initiation. One first phosphodiester bond is formed. So you can see here clearly, this is your first base, and once this binds, it is not called initiation. When second base binds to the second nucleotide and there is a formation of phosphodiester bond between these two first bases, then it is called as initiation. Then what is promoter clearance? Once this RNA polymerase move forwards by synthesizing continuously RNA, it will leave the promoter site. That is called as promoter clearance. That is when RNAP moves away from the promoter region. And elongation. So if continuously phosphodiester bonds forms between two bases, so randomly nucleotides comes and bind. If they match with the if they follow Watson grip carrying, the bond will be strong, so they will not go away. If it is A, C comes, it will go away. If G comes, it will go away. If A comes, it will go away. If T comes, it will bind strongly. So immediately RNA polymerase will form the phosphodiester bond. This will be in continuation process, and this process is called as your elongation. So your mRNA gets elongated, and now you should stop. So you can't continuously go on synthesis. It is waste of nucleotides, waste of time for RNA polymerase. So it should stop whenever it is necessary. If mRNA strand is completed, it should stop. As you saw in promoter region like Tata signals, there will be similar signals here. It is called inverted hyphenated signal. What is this inverted hyphenated signal? There will be a sequence like this. I am writing the same sequence in a straight form. When in this direction, RNA will be getting synthesized. When it gets free from the DNA, these two will bind like a hairpin. If you can see, this sequence is getting matched with this sequence and it forms a hairpin loop structure. So once this hairpin loop structure comes, your RNA polymerase will pass for a moment. When this pass happens, there is something called as termination factor. Sorry, it is denoted by rho. It is called termination factor. It can bind only to the passed RNA polymerases. So once the RNA polymerase gets passed by this hairpin loop, this rho factor comes and bind to this RNA polymerase, and it also has helicase activity. It separates the nascent strand, that is your new mRNA strand, from little will be still binding to the DNA, so it will separate that and it will dislodge the RNA polymerase. So what is the function of termination factor? It dislodges the RNA polymerase and it separates the newly synthesized mRNA strand from the DNA RNA hybrid. So this how termination happens. Now you have newly synthesized mRNA. This is nascent RNA. Which is synthesized in your nucleus. It can't go directly to cytoplasm. Why? Cytoplasm have certain enzymes called as exonucleases. So we'll see what is the difference, what is exonucleases. If this is your, either it can be a DNA strand or your mRNA strand. If what these exonucleases do is, it will directly goes and start chopping off your nucleotides. So what happens to your strand? It will be getting shortened and shortened due to this activity. So you should protect this, your new mRNAs from this. There is also, you would have heard, endonucleases. These endonucleases actually cuts in between. But for these endonucleases to get between, you should have some patterns, like as you saw for promoter Tata box, you should have like TC pattern to get recognized by endonuclease. But these patterns will be not present commonly, so we don't have any problems with the endonucleases. So our major concern is our exonucleases. So there are two types of exonucleases. One is 5'3' nucleases. The other one is opposite one, 3'5'. So 5'3' will be acting this direction, so it will chop off the nucleotides from this direction, whereas 3'5' will be chopping off your nucleotides from this direction. So if mRNA goes to cytoplasm without any protection, it will get chopped soon. So there is no use of synthesizing new mRNAs without utilizing for protein synthesis. So we have some protective mechanism. So what we have? So in 5' we have cap-like structure called as 7-methyl guanosine cap. So if this is present at the 5' end, these exonucleases can't act. So now we have protected from the exonuclease attack on one end. On second end, we have continuous poly-A structure around for 200 nucleotides. So once mRNA gets synthesized inside the nucleus, so this is our nucleus, it should exit through the pores into the cytoplasm. Before exiting through the pores into the cytoplasm, there will be poly-A polymerase enzyme. This poly-A polymerase will add continuously adenine nucleotides at 3' end. It doesn't require any template. That is the specialty, and this can be asked as MCQ in PG entrance exam. So which polymerase enzyme doesn't require any template for synthesis? That is your poly-A polymerase. Other all polymerases require a template strand to synthesize. But poly-A polymerase, it's just like that, can add continuously A nucleotides. It adds around 200 nucleotides and it drops off. So we have no protection at 5. So whenever this 3' ends, once exonuclease get cuts also, only A gets chopped off and it is not a big problem. Before the exonuclease reaches this end, your protein synthesis would have been happened. So this how you protect. This happens in both prokaryotes and eukaryotes. Something special happens in is splicing. So what happens in eukaryotes is coding and non-coding region both in mRNA. In prokaryotes, the mRNA that is synthesized can be directly utilized for protein synthesis. So whole thing will be coding for the protein synthesis. But in eukaryotes, there will be coding and non-coding region. The coding region is called as exons and the non-coding region is called introns. So we should remove this introns from the mRNA. So splicing will be done. What is this splicing? It is done by Spliceosome. What all consists of spliceosome? Your primary transcript, that is the RNA that is freshly synthesized. Your SNRP, small nuclear RNAs that I initially told, non-coding type of RNAs, and RNA recognizing protein, that is RRP. This all together will form spliceosome. What this spliceosome does is, how it will differentiate exon and intron. There will be certain sequences at this junction called consensus sequences. The same sequence will be present in this junction. So if there is consensus sequence, like in termination, if these two comes near, they will form a hairpin structure. So what this spliceosomes does is, first it will excise this site. So this site will get free, and afterwards it will excise this site. So when this site is excised, these two will form loop structure and it will go off. So this site and this site will get joined, and you will have all the exons getting joined like this. This is called splicing. Another application of splicing is, we'll come to that now. So there is one more major difference in prokaryote and eukaryotic gene. We'll take example, tryptophan metabolism. You know, we need many enzymes in synthesis of tryptophan. If you take prokaryotes, all the genes that is required for synthesis of tryptophan will be present on single gene, and it will be present continuously. That means nearby. So if mRNA is synthesized, all will be in the same mRNA, and once protein is synthesized, it will get separated. Whereas in eukaryotes, for one enzyme, it will be on one chromosome. For one enzyme, it will be on one chromosome. The other enzyme will be on another chromosome. The fifth enzyme will be on another chromosome. Third enzyme will be on another chromosome. So there will be different mRNA transcripts, and they will be synthesized differently. And sometimes the same mRNA transcript may have genes for 2 to 3 different enzymes also, which are unrelated. So that's where splicing will come. If these two will be joined, it produces one type of protein. If these two will be joined, it produces one type of protein. So this how it happens in eukaryotes. Whereas in prokaryotes, it is very simple. It just continuously synthesize all from one gene, and translation happens and proteins will be separated. Whereas in eukaryotes, you need different number of genes, and all will be differently synthesized, and proteins also will be differently synthesized. So that's all about transcription. If you have any doubts, you can ask me now. Hello? So everything is clear?
Yes, sir. Everything is clear, sir. Thank you.
Okay.
What about you, Anush Kumar? Clear?
Okay, so Akhila has asked, what is rho factor? It is a termination factor. I said it will be freely present in nucleus. It will be freely present in nucleus. Whenever this RNA polymerase passes, that whenever I said when hairpin loop structure comes in between, RNA polymerase will just pass for a moment. So when it passes, this rho factor can bind to this RNA polymerase. Why it can bind? There will be a structural or slight configurational change when RNA polymerase gets passed. So when there is configurational change in RNA polymerase, this rho factor can come and bind to RNA polymerase, and it will stop the RNA polymerase from synthesizing, and it will have helicase activity, which will separate your mRNA transcript from the from your template strand. It is mainly a termination factor to stop the mRNA synthesis or your RNA synthesis. Did you get it?
Yes, sir.
Okay.
Sir, what is the smallest RNA, sir?
Means which is present in smallest amount you are asking.
Smallest in size.
Smallest in size will be your siRNA.
siRNA.
Yeah, silencing RNA.
Okay, sir.
It will be like around 20 to 25 nucleotides also sometimes. And your longest will be either long non-coding or mRNA, that you can't tell specifically. If you have very large proteins, mRNAs will be very longer.
Okay, sir.
Okay. Any other doubts?
What is sense codon and nonsense codon?
Okay, that is actually I will be explaining in next topic actually whenever I take translations. Okay.
Okay, okay, sir. Thank you. So template and non-template, it is similar to sense and nonsense, or simply either
No,
different?
Here that is codon. Here you will using strand. If you can see now the screen, anti-sense strand and template strand are same, whereas non-template and sense strand are same.
Oh, okay, sir. Okay.
Okay. Any else doubts?
No, sir.
Okay. Thank you.
Thank you, sir.
Okay.
Thank you, sir.
Okay.
Yes, sir. Thank you. Sir, any kind of MCQs in this topic?
As I said, the most one important MQ is this will be how much percentage this rRNA will be there. That is one most important one. And again, which will not have proofreading in transcription, why there is no proofreading in transcription, you should know the reason. Next, and also there will be no primer for transcription. And one more thing I said, like similar to no primer, no template for poly A polymerase. These two are important. Then how you differentiate these three types of RNA polymerase, type 1, type 2 and type 3. And what is the promoter site? That is Tata box site in case of prokaryotes, and in case of eukaryote it is called as cat site. And you should also know that promoter region is extended also both in downstream region and upstream region in case of eukaryotes. This you should remember. And then you should remember when you call as promoter clearance and when you call that transcription has been initiated. Not just adding a first nucleotide, first phosphodiester bond should be formed, then you call it initiation. And when does rho factor binds actually, and what is the important function of rho factor? And there in this case of initiation, what is the function of sigma factor? I told you it will increase the affinity of binding, whereas rho factor it will actually decrease the affinity of binding and it will have helicase activity to terminate the synthesis. And you should know here, you should know about poly A polymerase. It doesn't require any template. This will be the major MCQs that will be asked.
Yeah, thank you, sir.
Okay.
Thank you, sir.